Hi, in this video I'd like to go over some specific examples of metal ions and the properties that are useful for biological systems. Here is a periodic table that shows the elements that are biologically relevant in natural systems. And here's a list of the roles that metal ions play in natural biological systems. And the metals that are in these roles are specifically chosen for their properties. So here are the properties of metal ions that are useful for biological systems. And I'm just going to start at the top of this list and go over a few examples and give you some details about how the metal ions work to serve the role that they are in. So metal ions are used for their charge in electrolyte conductivity and charge balance. And one nice example of this is how metal ions are used in nerve impulses for their charge. And here you see a cell body of a neuron, the axon, and the dendrites are not pictured, but they would be down here. And the nerve cell sends an impulse down the axon to the dendrite where neurotransmitters are released. We're not going to focus on the neurotransmitter part of this, but just on the signal propagation down this axon. So you may know that nerve signals send their messages down down an axon through action potentials. And a very simplistic view of how an action potential works is shown in these boxes. So when the cell is in its resting state, the membrane is polarized. And that means essentially that there's a high concentration of sodium outside the cell and a very low concentration of sodium inside the cell. So this difference in sodium concentration causes a concentration gradient, which basically is storing energy. At the same time, there's a high concentration of calcium inside and a low concentration of calcium outside. So you basically have two different things here that have very different concentrations inside and outside the cell, and they cause basically a potential across the membrane, an energy potential. So when a cell is going to send a signal down the axon, it does so by basically opening up the membrane for sodium to transport through. So there's actual channels in the cell membrane that allow sodium to pass down its concentration gradient, so the sodium will go into the cell and basically balance the difference in concentration here. And that's what's being shown in this picture down here. Here we know that outside the cell there's a high concentration. When you open up a sodium channel, it allows sodium to flow inside the cell and basically equilibrate the concentrations. So now instead of a higher concentration outside, you're equilibrating them, and now you're getting a higher concentration inside. What's not really shown in this picture is that after this happens, the membrane also has potassium channels that will open up so that the potassium ion concentration, which is high inside, will also equilibrate. And so this causes a polarization and it's polarization followed by depolarization and then repolarization of the membrane. I'm going to focus on the potassium channel because this is the one that's discussed in your textbook. And this is the figure from your textbook to show very simply what the potassium channel is. So you have a membrane that's polarized in the resting state, and it's depolarized as sodium concentrations are equilibrated. In response to a depolarization of the membrane, the potassium channel will release potassium. So just to kind of point out some important features here, which I don't think are really described well with this figure, but essentially you have a selectivity filter here and a cavity in this protein. The potassium ion here inside is shown to be in its aquated environment, so it's bound to water, and it's bound to water in this cavity. And then it needs to lose its water molecules as it passes out here. What else is being shown here is that the potassium channel can be in an open or closed conformation. Let's take a closer look. But first, let me tell you that the Nobel Prize in Chemistry in 2003 was awarded to Roderick McKinnon for his discovery and explanation for how the potassium channel worked. And he shared the Nobel Prize with Peter Agar, who discovered and explained how water channels worked in the membrane. Before their work, we didn't really understand how polar molecules or ions could transport through the cell membrane. And this is the paper that specifically discusses or reported the work on the potassium channel. So here's a crystal structure of a potassium channel in blue, embedded in a cellular membrane. So it's a phospholipid membrane that has polar head groups on the outside and the inside of the membrane. And the core of the membrane is a hydrophobic fatty acid tail core. So if a polar ion or a polar molecule or an ion wanted to pass through this membrane, there's a really high energy barrier for the ion. For example, an ion would be aquated or basically bound to several water molecules, and it's very polar, so it would have to pass through this non-polar region, and it might lose its water molecules in the process. And that's very energetically unfavorable, and it's simply not reasonable enough to happen. So we didn't really understand how an ion or a charged molecule could flow through this membrane. And the explanation is that there are ion channels, and for water there's a water channel, and for any polar molecule that needs to transport through the membrane, there's probably a channel associated with its transport. This is the potassium ion channel, and here you can see that there are 4 little green dots here, and those are potassium ions bound in this region here that we call the selectivity filter. This protein specifically transports potassium, and no other ions will go through this filter. As an aside, I'd like to talk about how this channel works, because it employs some of the fundamental inorganic concepts that we've learned in this class. So let's take another look at this selectivity filter. This is another view of just part of the protein. The protein channel is actually 4 subunits, and only 2 of them are shown here. So this channel was actually discovered using scorpion toxin, and the amino acids that interact with scorpion toxin are shown in white here. It's just interesting. This is just kind of an interesting aside. Scorpion toxin is a neurotoxin that prevents nerves from signaling to each other, and so it causes cardiac arrest in its victims. If you get stung by a scorpion, there's probably not enough toxin to hurt you, but if a mouse gets stung with a scorpion toxin, then the dose usually kills the mouse. And it kills the mouse because they have very similar potassium channels that are basically almost identical to ours. But anyway, the scorpion toxin blocks this pore, and so it stops cell signaling. The potassium selectivity filter is shown here in red. So 3 of the amino acids that are part of the selectivity filter are shown in red here. And those 3 amino acids are absolutely essential for potassium selectivity. And when I say potassium selectivity, I mean selecting for potassium over something else like a sodium ion. So this selectively and specifically transports potassium ions through the channel. And now I want to take an even closer look at the amino acids involved here. So notice that this is a GYG sequence. Here we've got the GYG sequence, and in this picture we're actually looking at 2 more amino acids that are part of this selectivity filter. GYG are the 3 essential amino acids for the selectivity of potassium, but the filter is made up of 5 total amino acids. Now, here the backbone structure of the amino acids and their side chains are shown, and again, you're only looking at 2 subunits. And notice that the carboxylate oxygens, so these oxygens of the peptide backbone, are actually lining the pore region. So you see 1 group of carboxylate oxygens here, 1 group here. The other 2 amino acids backbones are not shown, but their carbonyl oxygens are still represented by these little red dots. And inside of this selectivity filter, you see potassium ions. This is actually from the crystal structure of the potassium ion, and so you see the electron density of those potassium ions bound. So in the selectivity filter, there are always 4 potassium ions. Now, the coordination geometry of each potassium in this selectivity filter is kind of outlined by these red dotted lines. And each of the potassiums is in an 8-coordinate square antiprism environment. Here you see another potassium ion here at the bottom. And this potassium ion is part of the crystal structure, and it is aquated. So these little red dots here are oxygen from water. And this potassium is aquated in the cavity of the potassium channel. Now, if we look at the geometry here, this is also in a square antiprism 8-coordinate geometry. And basically what this selectivity filter is doing is it's basically mimicking the hydration sphere of the potassium in the selectivity filter. And so there's no energy barrier, or there's no energy difference between the potassium that's hydrated in bulk solution or the potassium that's hydrated in the cavity and the potassium that's in the selectivity filter. And that means that these two things are easily in equilibrium, and it allows the selectivity filter to pass potassium through at diffusion limited rates. So that's really, really fast. So let's take one more look at this a little bit closer, and I've just put some other structures here to show you what a square antiprism looks like. So the square antiprism environment that a potassium ion has in bulk solution in an aqueous environment is the same as a square antiprism that's found in the potassium ion channel selectivity filter. It's an 8-coordinate square antiprism. And part of the reason why Roderick McKinnon got the Nobel Prize for not only identifying but explaining how this works is that he explained why potassium can get through this channel, but a smaller ion like sodium can't. So here's a cartoon to kind of explain this. Here's a potassium ion bound to 4 waters. In real life, you would see 8 waters here, but we're kind of looking in 2 dimensions. So here you see the potassium ion bound to some water ions, or water molecules, and here's the sodium ion bound to water molecules. And notice the difference in size. The potassium ion radius is larger, and its hydration sphere is larger than a sodium ion. Down here, we're looking at the ions bound in the selectivity filter of the potassium channel. In the potassium channel, when potassium is bound, it has the same geometry, and the coordination sphere here is the same as if it were the hydrated potassium. So there's really no difference here in the hydration sphere versus the selectivity filter sphere. And so these things are in equilibrium and they have equal energy. On the other hand, the sodium coordination environment has to change. It has to lose its 4 waters, and when it's bound in the selectivity filter of the potassium channel, it's only big enough to basically bond to 2 of the ligands at a time. And so it has to change its geometry and its coordination environment to fit through the pore. And this, the sodium is more stable in its hydrated coordination environment than when it's bound to the potassium channel. And so there's a huge difference in the energy here, and so your equilibrium lies to the side where the sodium ion is hydrated. So this is an energy barrier for transport for sodium, which doesn't exist with transport for potassium. All right, so now we've seen how the potassium and the sodium can both act as charge balance in nerve signaling, but also we can explain how the potassium channel works and how protein channels in general can select for one ion over another just based on size. Let's talk about the geometry of metal ions. And geometry is typically used as a structural scaffold for protein folding. So here's an example. This is Calmodulin, and Calmodulin changes its tertiary structure upon binding 4 calcium ions. Calcium is used as signaling, and it's used as signaling through binding to proteins and changing its the tertiary structure of the protein. There are proteins on the cell surface that bind to or that recognize only the 3-dimensional shape of this calcium-bound Calmodulin. Now, calcium is used as a signaling molecule because it has very low concentration outside the cell. The concentration of calcium inside and outside the cell are tightly regulated through calcium channels or calcium pumps that will either allow calcium to flow through the membrane or pump it through the membrane. So outside the cell, typically, you have really low concentration of calcium, and at very, very low concentrations, you will find that the apo form of Calmodulin is the major species. But when a cell releases calcium, like so for example, when a nerve cell wants to signal another nerve cell, it will release calcium. And the local concentration of calcium between the, or in the synapse of the neurons will increase immediately. And as soon as that increase happens, the calcium ions will bind to the apo form of Calmodulin and create this calcium-bound form, which will then bind to the receptors on the other nerve cell. At the same time, these calcium-bound forms will be taken up, or calcium receptors will take the calcium up, and then the calcium concentration will decrease. And so basically, you're returning to this calcium, or the apo form of this Calmodulin. So the calcium concentration really fluctuates, and the labile nature of calcium is being taken advantage of here. Now, calcium being at such low concentration has kind of a disadvantage. So when you have really high concentrations of other ions, like zinc and sodium and potassium outside the cell, the question is, how does this Calmodulin protein select for calcium when calcium basically is the lowest concentration thing? And the answer to that is that these binding sites on the Calmodulin specifically select for calcium through hard-soft acid-base theory. And here we can see the calcium binding site in Calmodulin. It's a 7-coordinate site where the ligands are all very hard donors. So they're all oxygen atoms, and some of them have negative charges. So we see here that the binding site is made out of 2 or 3 aspartate carboxylate groups, a glutamate carboxylate group, and a carbonyl backbone, and a water. So this is a very hard ligand set, and it's in a 7-coordinate geometry. This selects for calcium, which is a very hard, Lewis acid, and calcium not having any d electrons has a very labile and flexible geometry, so it doesn't really have any preference for a specific geometry. And this is basically the perfect site for calcium. And so that's how this protein will select for calcium, let's say, over zinc, which is a little bit softer. Another example of structure, or metals serving in structural roles, are zinc finger transcription factors. So these are proteins that bind to zinc and use zinc to fold properly. Now, without zinc bound, these zinc finger sites can't bind to DNA. So here we see 2 zinc finger domains in a transcription factor acting as a structural scaffold to bind to DNA. And what these transcription factors do is they bind to specific sequences of DNA, and they allow transcription of DNA into RNA so that that RNA can be translated into protein. So without these transcription factors, you can't have expression of protein from DNA. And once again, you want these proteins to be able to select for zinc over other metals that are in the cell, and the way they do this is through coordination geometry and hard soft acid base character. So zinc is a D10 metal ion, and it doesn't have any preference for one geometry over another based on the ligand field stabilization energy, because there is no ligand field stabilization energy. So what the protein can do is it can adapt a coordination geometry that's disfavored by other metals. Most metals that have partially filled D shells will prefer some kind of octahedral, square pyramid, or square planar environment. And this protein selects for a D10 metal ion over others by providing a tetrahedral geometry. It also selects for zinc through hard soft acid base character of its ligands. So zinc is kind of a borderline to soft metal ion, more borderline. And so it uses borderline and soft ligands to select for zinc over other harder metals. All right, now let's talk about how Lewis acidity can be used, or an example. The classic example is carbonic anhydrase. This protein is using zinc in order to basically turn water into a potent nucleophile. So once again, as, and I showed you this in the last video, zinc binds to the water and it lowers the pKa of water bound to zinc, releasing a proton and creating a hydroxide ion bound to zinc. The protein also binds to carbon dioxide, and it uses the hydroxide ion to attack the carbon dioxide and create carbonic or bicarbonate. And then this is released and water replaces that ligand. This protein is really important for converting carbon dioxide, waste product of your cells, into bicarbonate, so that the bicarbonate can be transported in your blood, and then eventually it will be converted back into carbon dioxide when it reaches your lungs and it will be exhaled. Here's the binding site for zinc in carbonic anhydrase. So here, zinc again is a borderline soft metal ion. As far as biological metal ions goes, it's on the softer side of things. And here you can see it's bound in 3 histidine sites, so those histidines are nice borderline ligands. And there's also a water bound. This protein also hydrogen bonds to water, so that as soon as this water is converted to hydroxide, and then the hydroxide attacks the carbon dioxide, which is also held by the protein, this other water ion can come right in there and replace that. So this enzyme is not only taking advantage of zinc's Lewis acidity, but it's also taking advantage of the exchangeable ligands, so that it can exchange a water, or eventually a hydroxide, with another water. Let's talk about the partially filled d orbitals and how I'm going to give you an example of how a metal ion like iron is used for its interesting magnetic properties. A lot of the really interesting magnetic and electronic properties are seen in the deep sea life or in microorganisms. So I'm going to talk about magnetotactic bacteria. These are bacteria that are in just about every water source, so they're in fresh water, and they live in a transition zone between an oxygen-rich environment and an anoxic environment. What these little bacteria do is they use their proteins to biomineralize iron as iron oxides in these little nanocrystals that have a specific shape, and they line these nanocrystals up along the axis of their body so that they act as little compasses. And there's polarization here. So I'm going to show you kind of a little cartoon of how these magnetobacteria work. So these magnetotactic bacteria have a cell body, and they also have a flagellum. And they line their little iron nanocrystals up so that they orient with the Earth's geomagnetic field. And so it's funny because the bacteria that live in the northern hemisphere have their compass basically oriented away from their flagellum, whereas the bacteria that live in the southern hemisphere have their compasses oriented towards their flagellum. So they're actually oriented in different directions with respect to their bodies, their cell bodies, but they're both oriented so that when the compass is oriented with the Earth's geomagnetic field, when the bacteria are in the oxic or the oxygen-rich environments, so like high in the water where there's lots of oxygen, so near the surface, they'll turn their little flagella counterclockwise so that they swim down. And when the bacteria are too deep where there's not enough oxygen, they will turn their little flagella so they actually swim kind of backwards up into this oxic-anoxic transition zone where there's just the right amount of oxygen. The funny but kind of sad thing is if you take one of these bacteria from the southern hemisphere to the northern hemisphere, it will actually be oriented kind of upside down, and so it will die because it will swim the wrong direction. So if it's up here, it will actually swim up instead of down. So this is how biology has kind of harnessed the magnetic properties of iron for its own advantage. And this is also an example of biomineralization. So there are this is just kind of a cartoon that I made. Here's the flagellum and the cell body. These are single-celled organisms. And there are actually proteins that shape these nanocrystals in this little compass inside the bacterium. And there's an organelle called the magnetosome that where all of this happens. So very interesting chemistry happening here, but it's all protein-metal interactions that cause this. All right, now let's talk about redox and a specific example of where the redox or the multiple stable oxidation states of metal ions are used in biological systems. So before I told you that I gave you the example of iron in proteins, and we saw that iron's redox potential, or the iron 2, iron 3 redox couple, could be tuned from the regular aqueous iron redox potential of about 0.77 millivolts to a range of different potentials depending on the coordination environment and the donor ligands that are provided by the protein, or in some cases an iron-sulfur cluster. So the idea here is we're going to be talking about redox couples, and so that means that basically we're going to use a reduction potential, or basically the potential of an oxidized something to get reduced. And so in the case of iron, we're talking about an iron 3 going to an iron 2. So that's a reduction potential, and we use the reduction potential to talk about the redox couple, both the forward reduction and the reverse oxidation. So let me give you an example here. If you have a binding site that's really perfect for an iron 2, for example, it might look like this little picture here. When iron 2 is reduced to iron 3, the iron 3 is a smaller ion, and so in an aqueous environment, those ligands would contract along with the iron center, and you'd give off an electron. And then to go back to iron 2, the ligands would have to expand. So this is just a simple representation of how a protein can control the redox couple between an iron 2 and an iron 3. If the site itself is fixed in like a larger coordination environment, or it has softer ligands, it's going to prefer the iron 2, whereas a site that's smaller, or where the ligands are closer together, or maybe the ligands themselves are harder, would prefer iron 3. So if you have a binding site that prefers iron 3, it's going to make it harder to reduce the iron 3 to iron 2. And likewise, if you have a site that prefers iron 2, it would be easier to reduce iron 3 to iron 2. And so you can modulate the reduction potential, or basically how easy it is to reduce an oxidized form to a reduced form. And by talking about reduction potentials, we're we're talking about oxidation potentials as well. So how hard is it to oxidize iron 2 to iron 3, or another metal ion to a more oxidized form. So here's a figure from your textbook, and this figure is giving you reduction potentials of the things that are relevant to the electron transport chain. So if you don't know about the electron transport chain, it is it's a group of proteins in the mitochondria, or specifically on the membrane of mitochondria, that produce energy for your cell. So in your cells, sugars are metabolized to form ATP and NADH, which are reduced dinucleotides. And the electron transport chain actually takes the reduced NADH and it converts it into more ATP. And this is a figure that shows you the reduction potentials of the components of that electron transport chain, and many of these are metal centers. So on this diagram here, let's just kind of break it apart. Here you have the reduction potential, or the reduction potential of the couple. So, oxidizing, basically this is oxidation of a sugar. Here is the couple of a hydrogen ion and the H2. So this is the oxidized form and the reduced form. Here's NADH and NAD+. NAD+ is their oxidized form, and NADH is the reduced form. Cytochrome C, this is actually a copper center in cytochrome C that's going to change oxidation state. So for this, we're talking about copper 1 and copper 2. And here's nitrate and nitrite, and dioxygen and water. And oxygen here is your oxidized form, and water is reduced. So we're increasing reduction potential here. And what that really means, so down here at low reduction potential, it kind of, I take it literally, it's low potential for reduction. So these are things that don't reduce easily, or they're things that like to stay in the oxidized form of whatever couple that you see here. And then if it has a high if it has a high reduction potential for this redox couple, that means that it's more likely to go into the reduced form. It's more likely to accept electrons. So these are things that are very easily reduced, and that means they very easily accept electrons. And here it's showing that the electrons flow kind of up towards, or from the low reduction potentials to high reduction potentials. The ligands here, or the green bars here, are showing you the biologically relevant metal centers. So here we're seeing that iron-sulfur clusters can be tuned to span this range. We're seeing that cytochromes can be tuned to span this range. Copper centers can be tuned to span this range. And then we see manganese and iron centers up here, some iron 4 centers, that is. So iron-sulfur clusters are typically iron 2 and iron 3. And we also see some biological molecules here: sugars, hydrogen, NADH or NAD+. Cytochrome C is a protein that contains a copper center, nitrate and nitrite, and oxygen and water. And these are all players in the electron transport chain in the mitochondria. So what biology has done here is, if you want energy out of sugar, you basically need to get the electrons out. And biology has evolved molecules that have higher reduction potentials than sugar so that you can get the energy out of the sugar. And the electron transport chain is going to start with NADH. But again, these are metal centers. All of the things with the green bars are metal centers. And biology tunes the metal centers so that if you have an iron-sulfur cluster and you want to get your electrons up to oxygen, you can use an iron-sulfur cluster and bridge it with a cytochrome copper center, and then the electrons will jump up to oxygen and reduce water. You tune potentials using the first coordination sphere of the metal ion, so that's the actual donor atoms and the geometry. Remember that each metal ion will have a preference for a specific geometry and for a coordination size. You can also do this with hydrogen bonding interactions around that first coordination sphere. And the presence of neighboring charges can contribute, because when you oxidize or reduce a metal center, you're changing the charge of the metal, and the presence of neighboring charges can influence the stability of a higher charge or a lower charge. These are the things that affect the potential of the metal center. In this electron transport chain, you also need kinetics to work on your side, so these things need to happen very fast. And the kinetics depend on small reorganization energy. All of these electron transfer events are outer sphere electron transfer. And so basically you need for a metal center to be able to cycle through oxidation states, you need it to not reorganize too much. If the protein has to completely change its coordination environment for one oxidation state versus another, it takes time. And so you don't want to have to do that. You want a binding site that's going to accommodate both an iron 2 and an iron 3, and sometimes that means that they don't fit perfectly for either one of those things. They're kind of an in-between binding site, which allows the flexibility of the oxidation state of whatever metal you're dealing with. When electrons have to hop from one metal center to the other in an outer sphere reaction, the distance between the metal centers is important. And solvent polarity can affect the kinetics, and this is related to the small reorganization energy. Polar solvents must rearrange in response to a change in the charge of the metal center. And so polar solvents can affect basically the amount of reorganization that can happen, and they can change the kinetics and make things either slower or faster depending on what solvent you're dealing with. So make sure you read a little bit about redox in your textbook, and I will also post a video separately on the redox material. Here's a sketch of the electron transport chain. So this is basically after the sugars have been oxidized and NADH, which is one of the energy molecules of the cell, have been created. And so let's take a look at this diagram. So we have different proteins, and they're called complexes A, B, C, D, and E. So complex A is an NAD dehydrogenase. It's an iron-sulfur cluster, an 8 iron-sulfur clusters more specifically. Complex B has another iron-sulfur cluster. Complex C has another iron-sulfur cluster. And these guys, these are all iron-sulfur clusters whose redox potentials have been tuned so that electrons will flow from A to B to C. And so this sketch is showing basically NADH is oxidized to NAD+. I think that H shouldn't be there. And basically it gives off an electron in the process and a hydrogen ion. And the electron basically is now in an iron-sulfur cluster. And then it's transferred to another iron-sulfur cluster that has a higher reduction potential. And then once again to another iron-sulfur cluster that has a higher reduction potential. So if we took that little chart that I showed you in the last slide and we kind of tip it on its side and line it up here, basically your body started with sugars. It eventually produced this NADH, and that has an electron that can flow to an iron-sulfur cluster of the appropriate. So as long as the iron-sulfur cluster in complex A has a higher reduction potential than NADH, then the electron will flow to that iron-sulfur cluster. So we're increasing in reduction potential, and then at this point it seems like we might be at the top of the potential limit for iron-sulfur clusters. And so look what we have now in complex D. This is cytochrome C oxidase. This is a copper center, and a copper here accepts an electron. So here we've got an electron coming all the way from NADH through complex A, through complex B, through complex C. These are all iron-sulfur clusters. It then transfers to complex D, where it is accepted by a copper 2 and forms copper 1. And then now we're in this copper region, and the next thing that happens is that electron is accepted by oxygen, and oxygen is reduced by adding hydrogens here and 2 electrons to give you water. So here we can take NADH all the way, the electrons all the way down here to complex C, and you produce water from oxygen. And so this is respiration, right? We know that we breathe in oxygen, we use it for energy, and we breathe out water and carbon dioxide. And this particular thing doesn't show you where the carbon dioxide comes from, but at least you can see how part of it happens from this diagram. So here you see, you need a range of reduction potentials, and biology has evolved so that they tune these reduction potentials so that electrons can be leveraged and be funneled basically to where they need to go to produce energy for your cells. All right, now let's talk about exchangeable ligands. And the classic example for this is oxygen transport. So here's a picture of myoglobin, or one subunit of hemoglobin, depending on what you want to look at, or however you want to look at it. They look very similar. The porphyrin ring here is binding to iron, and in the apo form, where oxygen is not bound, the iron is iron 2, and it's high spin. So it has electrons in its T2G and its EG set, so it has electrons in the higher energy orbital of the DD splitting. And it's a larger ion when it's in this kind of a configuration. when oxygen binds here, what happens is now oxygen is a pi-accepting ligand, and so it actually converts the iron from high spin to low spin. And the low spin now does not have any electrons in its Eg star set, and so it actually kind of gets a little bit smaller. And as it does this, it's pulled up into the plane of this porphyrin ring, and being pulled up like that actually pulls the histidine. And this causes a change in conformation of the protein, which then basically in the next subunit of hemoglobin, there would be another one of these, and the histidine essentially would push on the apohemoglobin, the apoporphyrin, and it would like increase its affinity for oxygen. This oxygen is also hydrogen bound by this histidine here, and the iron again is low spin. Now, this is a hotly debated topic in bioinorganic chemistry, and that is the actual oxidation state of this iron. Once oxygen binds, you can either count this as an iron 2 dioxygen, or if you consider that maybe one of the electrons is transferred from the iron to the oxygen, you could call it an iron 3 superoxide. And most of the literature thinks that it's an iron 3 superoxide, but there's definitely a body of scientists that consider this an iron 2 dioxygen. These two situations are isoelectronic, so it's really just a matter of where is that electron. In either case, know though that the oxygen-bound form is low spin, and this should make sense to you based on what you know about the spectrochemical series. Once this oxygen binds, it's a good pi acceptor, and it increases this d orbital splitting of this iron center. So it's low spin when oxygen is bound, whereas it's high spin without oxygen. When oxygen comes off here, it just comes off as dioxygen. It never comes off as superoxide. Now, I mentioned the cooperative nature of hemoglobin. Once one of these irons adopts the low spin geometry, where it basically tugs on this histidine, it causes a conformational change. Now, this is basically, it's kind of like 4 subunits of myoglobin, but there's an alpha subunit and a beta subunit, and they are slightly different, but all 4 of them have heme. And here's a little table from your textbook again. So you can see that as you increase the partial pressure of oxygen, the percentage of the myoglobin, which would be like just one of these subunits, it increases linearly, and then it levels off. Now, there are 2 curves for hemoglobin that are shown. This blue curve at pH 7.6, this is relevant to the lungs, or the pH near the lungs, and the pH anywhere near the muscles, or more like away from the lungs in your body, the pH is more acidic. And when the pH is acidic, that means that your muscle cells need oxygen. So near the lungs, so you can see if you compare it to myoglobin, hemoglobin, it doesn't bind to oxygen as well. The first oxygens don't bind as well anyway. Once the first oxygen binds, though, then now you have a dramatic increase, and then it levels off. And what this does basically is you have to have a high concentration of oxygen for hemoglobin to bind any oxygen at all. And that's so that hemoglobin only binds oxygen strongly near the lungs, where the oxygen concentration is high. And then it'll bind a bunch of oxygen. And when the pH drops, now you have basically a lower affinity of hemoglobin for oxygen, and so it will release its oxygen, and that way your muscles can get the oxygen from the hemoglobin. And the myoglobin is the oxygen carrier in your cells, and so that has a much higher affinity for the oxygen, and so that's how basically oxygen gets from your lungs into the hemoglobin, and then the hemoglobin goes around to your muscle cells, it releases the oxygen and transfers it to myoglobin. The biggest difference, or the difference I wanted to point out here, is that this is basically a linear increase, and this is a sigmoidal increase. Hemoglobin and myoglobin are the oxygen transport molecules in mammals. In arthropods and mollusks, we have copper binding centers as the oxygen carriers. So this is hemocyanin, or the binding center in hemocyanin, which again is in these blue crabs, or crabs and mollusks. And basically a dioxygen molecule will fit right between those two copper ions, and it will bind to copper, and then it can be released. So now we've seen some examples of the metal ions that are playing some important roles in biology based on their specific properties.